Video 4 fills in some of the background information about NADPH. We have looked at the similarities between the cytokine storm in radiation and in COVID-19, recognizing that ketone ester was able to save the majority of lethally exposed mice. We have looked at how SARS-CoV-2 attacks the AEC2 cells and the loss of 4 functions. We've looked at how SOD3 and catalase work as true scavengers due to their dismutase reaction mechanisms to clear superoxide and hydrogen peroxide. We also looked at how antioxidants that are not dismutases work. In looking at antioxidants, we noticed that the antioxidants must get an electron from some source in order to be reused. That source is ultimately NADPH. In the COVID-19 picture, R3-hydroxybutyrate converts NAD+ to NADPH. Under certain conditions that need to be better understood, R3-hydroxybutyrate or beta-hydroxybutyrate, BHB, creates greater or more negative redox potentials of NADPH in ketosis than in a well-fed state. That greater redox potential of NADPH shifts all of these roles of NADPH in a positive direction as it relates to COVID-19. NADPH is required for maintaining antioxidant defense. It regulates cortisol activation and deactivation. It's required to kick off the production of ROS and RNS as part of the nonspecific immune response via NADPH oxidase. It regulates transcription factors NRF2 and HIF1 alpha that are master regulators of the antioxidant response elements and hypoxia response elements, respectively. It modulates redox-sensitive chaperone DJ1, which senses hydrogen peroxide as a signal. Mitochondria clearing of the hydrogen peroxide. Fatty acid synthesis. Neurotransmitter synthesis, adrenaline synthesis, mitophagy, autophagy, apoptosis. What is NADPH? The D stands for dinucleotide. There are 2 nucleotides. So, what is a nucleotide? Nucleotides are the building block or individual monomers that make up the polymers RNA and DNA. Nucleotides consist of 3 parts: a nitrogenous base, a 5-carbon sugar, and a phosphate group. The 5-carbon sugar is ribose in the case of RNA and deoxyribose in DNA. Nucleotides also function as coenzymes such as ATP, coenzyme A, NADH, and NADPH. The P in NADPH refers to a phosphate group that is attached to the 2' position on the ribose sugar of AMP. The 2' phosphorylation of adenosine monophosphate allows 2 sets of enzymes to use the NAD and NADP for energetically opposite uses in the same compartment. How is NADPH related to beta-hydroxybutyrate? If one looks in any biochemistry book, there is an explanation that the redox potential of the cytoplasmic NAD differs from the redox potential of NADP. The relative ratios of NAD+ to NADH and NADP+ to NADPH differ by a factor of 10 to the 5th. The reason we know this is due to Dr. Veitch and Hans Krebs. Dr. Veitch was assigned the task of measuring the ratio of NADP+ to NADPH in the cytoplasm by Krebs in response to a suggestion of George Cahill. Dr. Veitch found that NADPH has the lowest, that is, most negative redox potential of any of the major biomolecules. This positions NADPH to be the ultimate source of all the electrons that are used to reduce antioxidants. Dr. Veitch also found that fasting animals have a lower NADPH redox potential than well-fed animals. This held true for animals fed ketone ester, as long as the diet was not high in carbohydrates. The lower redox potential and the production of NADPH through metabolites of the Krebs cycle that leave the mitochondria ties R3-hydroxybutyrate to NADPH. This figure shows many of the pathways that allow the electrons from NADPH to reduce spent antioxidants. In this video, we looked at NADPH and how it is needed to allow antioxidants to clear the increased load of reactive oxygen species and reactive nitrogen species that generate damage-associated molecular patterns, or DAMPs. In turn, the DAMPs keep the innate immune response making more ROS and RNS. In the next video, I want to examine NAD and NADP and talk about how they affect the cortisol-cortisone relationship.